Welcome to a very exciting webinar today by Dhriti. So let me just introduce Dhriti. Dhriti is a postdoctoral fellow at the Sydney Brenner of the Institute for Molecular Bioscience here in Johannesburg. She obtained her PhD in bioinformatics from the University of Calcutta in India. And Dhriti's main interests are inferring fine-scale population structure, understanding migration patterns and population interactions. And she has been a key analyst for some of the major AWI-Gen and H3 Africa population genetics and disease association studies. Dhriti, thanks for accepting the invite for the webinar, and we can't wait for your talk. But briefly, Dhriti just wants to say hello before we can start.
Thanks, David. And I think all the participants I see here, pretty much everyone knows me. So I was not very sure about my internet connection, so I just recorded this talk. And I think once David has played this one, and then I will see you everyone on the other end of the question and answer session. And thanks, David, for this opportunity.
Awesome.
Good evening, everyone. It's indeed a pleasure for me to present my research in this forum. Today, I'll be talking on the largest genetic study from South Africa. I would begin by recapitulating the current knowledge about the peopling of South Africa, that is the history of arrival of diverse groups of people that make this country a rainbow nation. Unlike many other parts of the world, you will find the history written, actually painted on the rocks over Southern Africa. The only people who lived here for many tens of thousands of years probably consisted of the ancestors of the present-day Khoe and San hunter-gatherers. Although there were a few smaller migrations, the event that changed the demography of this country, of this region, was a conspicuous large-scale migration of Bantu-speaking agro-pastoralists. The Bantu expansion started from the borders of Nigeria and Cameroon around 5,000 to 4,000 years ago and reached the Great Lakes region in Eastern Africa by 3,000 years ago and South Africa around 1,500 years ago in distinct waves of migration. And finally, around 400 years ago, Europeans started arriving via sea. Coming back to Khoe-San, which is a kind of catch-all term for all the indigenous people of Southern Africa. As demonstrated in the figure at the left, you might already know the ancestors of Khoe-San were the first to diverge from the anatomically modern humans. That was followed by the divergence of the ancestors of rainforest hunter-gatherers. And finally, the split of the East and West Africans all happened within the continent. The ancestors of modern Khoe-San expanded to Southern Africa before 150,000 years ago, possibly as early as before 260,000 years ago. So they were once spread across most of the sub-Saharan Africa, but now they are unfortunately limited only to a region around the Kalahari Desert. The Bantu expansion was perhaps the most crucial large-scale migration that happened in the history of the continent that reshaped the genetic landscape of Africa. The Bantu speakers moved towards the south in different waves, some along east and some along west. The Bantu speakers arriving on the eastern side gradually spread across present-day South Africa between 400 to 1300 CE. There is a known history of interactions between the Bantu speakers and the Khoe-San populations, which along with other historical factors have given rise to different Southeastern Bantu-speaking linguistic groups, such as Zulu, Sotho, Xhosa, and Tsonga, etc. The map on the right shows diversity of the first language in the country. These images are intended to showcase the cultural diversity that parallels the linguistic diversity of various Southeastern Bantu speakers or ACB groups. While there has been quite understandably a lot of interest of the Khoe-San linguistic and genetic diversity in Southern Africa, the Bantu speakers did not receive proportionate attention. And in spite of the linguistic and cultural diversity, the genetic differences between these groups have not been investigated systematically, and these are often considered as a homogeneous genetic unit. The first study to provide the view that there might be some structure within the ACB groups was published in 2002. The study included all the main ethnolinguistic groups and was mainly based on Y chromosome and few autosomal markers. With the help of neighbor joining tree, they were able to show that the two Nguni speaking groups, Zulu and Xhosa, form a branch. Similarly, geographically closer Songa and Venda also share higher genetic similarity compared to others. So we could see higher proximity of the geographically or linguistically closer populations. Although many later studies, some examples shown here, used genome scale data, did not replicate or report this structure. In 2017, while working on the Southern African Human Genome Program study, we were able to see that there is a subtle structure between Sotho and Xhosa, and found that there is a difference in the Khoe-San ancestry proportion in the two. However, this study was based on 7 individuals per group only. So there was evidence, but not conclusive enough. Based on the largest genotype dataset from South Africa, including more than 5,000 individuals, we aim to investigate systematically and deeply the genetics of these ACB groups. Our participants were sampled across 3 sites from northeastern South Africa. These participants were genotyped on the H3 Africa genotyping array. The dataset represents most of the major ACB groups from South Africa. The corresponding numbers are shown below the names. So the main question that we asked are, is there a population structure and some level of genetic differentiation between these groups? If yes, why does the extent of structure vary between different studies? And what is the source of this difference? And could this structure have any implication on biomedical studies? So first we investigated if there is a population structure within the ACB groups. And the first analysis of any PopGen study is perhaps PCA, where each individual is represented by a dot and each population is represented by same color. The primary population structure analysis performed using PCA shows intrinsic diversity in the unrelated ACB individuals. The PCA showed some reflection of the ethnolinguistic classification with rather partial separation between the 3 major ethnolinguistic groups consisting of Songa, Sotho-Tswana, and Zulu and Xhosa. The PC1 splits Songa, shown in cyan, from remaining ACB groups, while PC2 splits Sotho-Tswana speakers, shown in green and red respectively, from Nguni speakers, Zulu shown in blue and Xhosa in orange. Interestingly, the distribution of various ACB groups in the PCA was found to mirror the majority geographic locations of these populations, shown in the same color on the South African map. However, the separation between the ACB groups is only partial, with considerable overlap between some of groups. Moreover, in some cases, individuals from one group cluster with individuals from other group. Since we had the ethnolinguistic label information from not only the participants, but also their parents and grandparents, we thought of going back to this to get more information on the individuals who clustered with different groups. And so, we found something really interesting. A significant proportion of the individuals had ethnolinguistically concordant ancestry, that is, having all parents and grandparents with same ancestry. Then, of course, we had many individuals with mixed ancestry, mixed ACB ancestry. Intermarriages between various groups is common, especially in urban areas. And as you would have guessed, many of these individuals who had multiple ACB ancestry were multiple ACB ancestries made them appear between the clusters rather than within the cluster. So we generated a subset of ethnolinguistically concordant individuals only per ACB group. So here is a table with all the details for each ACB group per site. The first column here shows all the individuals, and the second column shows the number after retaining EC individuals only. The subset with EC individuals was much smaller, that is only 2,700 out of the around 4,500 individuals. Groups such as Swazi and Venda were reduced by about threefold. And other than Songa, where most EC individuals were from Agincourt, and Pedi, where most EC individuals were from Dikale, the maximum number of EC samples were from Soweto. Although Soweto is the melting pot, you have to remember that the individuals here are between 40 to 60. So the population we see here is actually 2 generations ago. It may be very different if the study is done on the individuals who are much younger. The PCA regenerated with EC samples noticeably enhanced the resolution of different ethnolinguistic groups and reduced the number of participants who cluster with different ACB groups. This time, we clearly observe existence of at least 3 distinct genetic groups corresponding to Nguni, including the Zulu, Xhosa, and Swazi, the Sotho-Tswana group, and Songa, the main linguistic groups among the ECB. This was very interesting as such distinct clusters were never observed in the previous studies. Okay, so this plot is based on 2 PCs only. Now, as you might know, the more PCs in a plot, the more information you get. The PCA map plot on the right is a composite representation of the first 10 PCs, and it makes the separation of the ACB groups much clearer. Next, the AWI-Gen dataset was merged with previously genotyped populations from neighboring geographic regions. This PCA is based on different ACB groups from the African Genome Variation Project, South African Human Genome Program study, and few other studies. We observed overall concordance between the populations from different datasets. The ACHGP and AGVP Sutu cluster with the AWI-Gen Sutu, all shown in green with different symbols. Similarly, AGVP Zulu, shown in sky blue, localizes with AWI-Gen Zulu, shown in navy blue. And the set of samples from recent Mozambique study localizes with Songa. And just in case you were wondering if the structure we are seeing is an artifact of the disproportionate sample size, this is a PCA with the AWI-Gen samples downsized to around 80 EC individuals for each group. The plot on the right shows a haplotype-based PCA of the ACB groups from the downsized subset. The haplotype-based PCA was estimated on the basis of co-ancestry matrix using fine structure. Both the SNP-based and haplotype-based PCA plots are identical. The population structure, as you can see, still persists. So disproportionate sample size does not seem to be a big factor here. As hinted previously, we observed that the PCA is by and large concordant with their geographic locations. On the top left, you can see the main geographic locations of the ACB groups from our study. And below, you can see the PCA downsized, PCA of the downsized individuals rotated to match the geography. The correlation is around 0.72, which is pretty good. We also observed that the UP geometry based on pairwise FST distance between the ACB groups from our study concurs with the topology of the independent linguistic phylogeny. The Nguni and the Sotho-Tswana clusters are clearly evident here. Now that we have clearly shown that there is a population structure within the ACB groups, we wanted to investigate if there are differences in ancestral profiles. That is, if these groups have differential gene flow from non-Bantu speaking ancestry. This is the admixture plot showing all the ACB groups along with selected Khoe-San and other populations for contextualization. The ACB groups were once again downsized to make the number of individuals per group similar. As you might know, in an admixture plot, each line is representing an individual and each color a distinct as ancestry estimated using the algorithm. In the plot, the case ancestry is shown in green, Bantu-speaking ancestry in blue, and Eurasian ancestry in red. In line with the previous studies, we observed the most ACB groups have significant proportion of Khoe-San gene flow. However, there are differences in the level of Khoe-San ancestry among the groups. Swana having the most Khoe-San ancestry contribution, around 20%, while Songa having the least, around 2%. We also observed that Zulu and Sutu from AGVP show very similar profile with our dataset. Now, let us focus on some more nuances. The admixture plot at the top shows all the Zulu, Sutu, and Swazi samples. And in addition to the ethnicity, the site of collection is also added to the information. In all the three cases, what you see is individuals from Agincourt, even though fewer in number, have lower Khoe-San ancestry in them compared to Soweto counterpart. The plot at the bottom shows the Khoe-San ancestry proportion differences between the individual samples from Soweto and Agincourt for the different ACB groups. The differences were statistically significant. The PCA plot on the right also reflects the same. Just like we did for the admixture plot, each individual here is labeled by the ethnolinguistic identity as well as sampling site. These are few ethnolinguistically concordant Zulu and Swazi individuals when sampled from Agincourt cluster with Songa individuals who were predominantly sampled from Agincourt. So basically what this tells is that consideration of ethnolinguistic identity is not sufficient and geography matters. This table shows all the overall ancestry proportions of the ACB groups and the differential level of Khoe-San and Eurasian gene flow into them. Interestingly, admixture estimates demonstrated the existence of more than 5% Eurasian-like ancestry in about 170 of our individuals, of our samples here. As there has been no systematic study to estimate the number, to estimate the level of Eurasian ancestry in the more northern provinces of the country, we had no comparative mechanism to estimate whether the observed level of Eurasian ancestry is common in these geographic areas or was something cohort-specific feature. This could provide a baseline for future studies on Eurasian admixture in the ACBs. The differences in Khoe-San ancestry proportions for the different ACB groups is also seen clearly. As we know, differential gene flow could result in the population structure. We wanted to know, or rather we wanted to investigate if that was the scenario in our case. For that, we masked the Khoe-San ancestry regions from the genomes in each ACB individual and performed the PCA again. The rationale being that if Khoe-San gene flow is the sole reason for the observed structure, masking any non-Bantu ancestry would cause all the ACB groups to collapse into a single PCA, like single cluster in the PCA. Identifying the ancestry-specific regions could be done using a technique, local ancestry, which basically infers the ancestral origin of a particular locus in the admixed population. The representative figures show the local ancestry deconvolution for the 22 chromosomes for a Swana and a Songa individual, where the blue segments represent Bantu speaker ancestry and red represent the Khoe-San ancestry. We performed ancestry-specific PCA with the genomic regions from Bantu-speaking ancestry only and observed that the core differences between the ACB groups, although reduced, persist even after accounting for the differential Khoe-San gene flow. Now, this hints that the observed structure between the ACB groups could therefore be a consequence of other demographic factors as well. One such factor could be differences in admixture timing. Admixture dating can infer a lot about the history of a population. If you have a resident population at a given geography and a new population moves in and mixes with the existing population, the descendants of the admixed population would carry footprints in the DNA that can help us estimate the approximate timing of the admixture event. And more sophisticated methods can also infer if the admixture event occurred in one or more, like multiple pulses. To delineate the timeline of migration of Bantu speakers, we dated the admixture between the Bantu speakers and Khoe-San source populations for each of the ACB groups using different techniques, like fast globetrotter shown in red and Mulder shown in blue. Our dates overall correspond to the previous studies, but provide a more granular estimate of the admixture. The Songa shows the oldest admixture date, around 45 generations ago, while the same for other groups like Swana and Khoza are around 25 generations. This is also supported by archaeological evidences where which indicate that the Songa might have arrived well before the Nguni speakers. So this suggests the spread of Bantu speakers was a complex process which could have involved multiple waves of migration or punctuated expansion. The recent availability of ancient genomes from Africa provides us with a unique opportunity to compare the affinities of present-day ACB groups with the ancient Bantu-related genomes. The Iron Age genomes showed a climb from Songa to Sutuswana and Nguni speakers that aligned broadly with geography, that is from north to south, and the time, that is the older genomes closer to Songa, as well as aligns with the increasing levels of Khoe-San ancestry in them. Now, more ancient genomes from Southern Africa would be required to test whether the trends observed in these 5 Iron Age genomes are indicative of phases in the movement of the groups further south with time, a process marked by the concomitant increase in the Khoe-San ancestry in the migrants. We were also interested in learning about the demographic histories of the ACB groups. So when two groups meet, there can be different kinds of interaction. For example, in one scenario, you would have males and females from each group marrying one another at a nearly equal proportion. However, in history, this scenario is rare. It was always the more powerful group got females from the other group, but the opposite was rare. That is, the bias of sex in mating is often an indicator of the nature of interaction and social dominance. We were interested in studying whether there was any sex bias between the interaction of the Khoe-San and Bantu speakers. Our study based on mitochondrial DNA and Y chromosome haplogroup distributions, as well as comparison of X chromosomes and autosome contributions, demonstrated a clear sex bias gene flow between the Bantu speaker males and the autochthonous Khoe-San females for each of the ACB groups. However, the extent of the Khoe-San maternal bias was observed to be highly variable among the ACB groups. This suggests that despite an overarching trend, these interactions were strongly influenced by locally defined factors that were unique to each of the interactions. Another key component of understanding the history of a population is to follow how the population size has changed over time. So this can inform us about the population, like how the population reacted to past events such as climate change, large-scale migrations, glacial cycles, and so on. We used a program IBDNE to investigate the changes in the effective population size of the ACB groups represented by each line in the last 100 generations. The shaded area show the confidence interval of the estimates. As evident from the plot, the NE, that is the effective population size for all the ACB groups, were very similar for the initial 60 generations, and the individual groups only began to diverge from around 40 generations ago. This matches with the generally accepted model of Bantu expansion, which suggests that sharing of a common ancestor of all the present day ACB groups until that time range. Moreover, if you remember from the previous slide, this period of 40 generations ago also corresponds to the oldest Khoe-San admixture dates in our study. We also observed distinct and characteristic Eni profiles for the Nguni speakers, here Zulu and Xhosa, and the Sotho-Tswana speakers. Interestingly, the dates for the initiation of the population size increase of the Zulu around 25 generations ago broadly correspond to archaeological records for the movement and settlement of Nguni speakers in certain regions of South Africa. A key question in this regard, which we were worried about, was that while we are finding interesting trends, are these estimates robust? Like, how seriously we can take them? To assess the robustness of the estimates, we performed a comparison of the Eni profiles for the Sotho and Zulu from our study to the Sotho and Zulu from the AGBP study. The results show similar timescales for separation and a decent concordance in the Eni fluctuations thereafter, suggesting that the observed trends in the Eni dynamics have a good chance of being related to the actual demographic history of these populations. Now, we know that admixture could potentially impact any sophisticated popgen analysis. The ACB groups, as we have seen, have varying levels of admixture happening at different points of time. So the Eni dynamics that we are seeing here not only relate to ancestral Bantu speakers, but also ancestral Khoe-San population. And for making a meaningful historical inferences, partitioning of the contribution of the different ancestral populations was critical. Therefore, we used an approach, ancestor-specific IBD-Eni, to achieve this. We generated Khoe-San and Bantu speaker ancestry-specific Eni profiles and compared them to the Eni profile of each of the ACB groups. The two plots show the ancestry-specific profiles for Tswana and Tsonga. The black line here shows the Eni for the whole population, that is the overall Eni. The orange shows the contribution from the Bantu speaker ancestry, and blue shows the contribution from the Khoe-San ancestry. In both cases, we observe that the Bantu speaker ancestry specific Eni predominantly guides the overall Eni in the last 40 generations. Similar trends was observed for the other ACB groups. Okay, so I hope I have been able to convince you that there is a clear population structure which informs history. But some of you might turn around and ask, like, so what? Why should it matter? Well, it matters if the observed structure impacts phenotypes and genetics of traits and diseases. I will now take you through some investigations into the larger impact of the observed structure in the biomedical genetic studies. The plot on your right shows allele frequency variation of some of the well-known phenotype associated variants in the 6 ACB groups. An interesting example here is the APOL1 variant associated to resistance to Trypanosoma rhodense that shows almost 6-fold variation between the groups, with the lowest frequency being 3% in the Tsonga and the highest frequency in Xhosa around 18%. The notable variation in the frequency of this variation in the major ACB group suggests that one group would deal with trypanosomiasis 6 times better than the other, or in a converse way, one have lived for a long time in a sleeping sickness dominated geography while the other didn't. So we observed more than 900 SNPs with high allele frequency differences within ACB groups that are associated to known phenotypes. Therefore, a public health intervention based on one ACB group could be quite misleading for the whole population. We were also interested in exploring the possible impact of this structure within the ACB groups on genome-wide association studies and the candidate gene-based studies as well. As most of you here know, genome-wide association study, or GWAS, is an approach used in genetic research to associate specific genetic variants with a particular disease. The candidate gene-based studies, on the other hand, test for one or few previously found associations between genetic markers and the phenotypes of diseases. This is often done in a population different from the discovery cohort. To investigate the possible impact, we generated 4 sets of simulated case control scenarios. In each scenario, we used 3 sampling sites and the different ethnolinguistic group composition as artificial traits and performed GWAS analysis on each set. So here is an example of simulated scenario 1, where 800 randomly sampled individuals from Agincourt, mostly including Songa, were treated as cases, and 800 randomly sampled individuals from Soweto, including multiple ethnolinguistic groups, treated as controls. The plot on your right shows representative QQ plots for this scenario. As you can see, the structure impacts the QQ plot quite significantly. That is, in an ideal plot, the black line should have been very close to the red line, thus making this a copybook bad data set for GWAS. But we wanted to make it more realistic, so we ran a few more simulated trait GWAS, which has a mixture of cases from the 2 centers and controls from Soweto, as well as taking all the cases and controls from Soweto with unequal representation of ethnolinguistic groups. Here are some of the examples of QQ plots from such scenarios. I'm not going into details, but you can clearly see in some cases, the impact of population structure is very strong, while in some cases, although the impact is at a lower scale, there is still a hint of structure. That is, there is an early divergence from the expected line. Now, those of who you are like working with GWAS would argue that there are ways you can correct for the population structure, and we did that. So you can see that the AGT Soweto, like the Agincourt Soweto QQ plot, there is a post-genomic control adjustment and correction that is shown in the green, and the correction using the first 3 PCs as covariates shown in pink. Both the approaches seem to control the structure. The PC-based approach seems to do better than the other one. However, there is a small but here. The table shows the stats of 50 simulations for scenario 1. Only the signals that appeared in 10 or more runs are listed. It produced thousands of genome-wide suggestive hits, and all basically false positive hits, probably reflecting the inherent structure in the ethnolinguistic group from the 2 sites. While the adjustment largely worked, there are still many false positives, especially at a suggestive level. Moreover, while the correction worked for within site and mixed site cases, they failed to address problems in cases with lower genetic distance, such as within Soweto comparisons. I guess we need more investigation on this. Therefore, while the broad brush correction works, they are not fail-safe. So while performing a GWAS analysis on ACP groups, caution should be taken to correct the population structure, and especially in case of candidate gene-based studies, where population-based correction is not possible, extreme care is needed. So finally, the take-home message. We have conclusively shown a clear population structure with 3 genetic clusters corresponding to the main linguistic groups in ACB. Khoe-San gene flow seems to be a major source of the observed structure. However, the structure also clearly corresponds to differences in the admixture dates and other aspects of demographic history. The population structure is strong enough to influence GWAS, especially with samples from multiple geographies and ethnolinguistic groups. And overall, the study showed the importance of proper population genetic characterization of understudied populations in informing both history and disease studies. So this brings me to the end of my presentation. I would like to thank the organizers, my supervisor, my funder, my colleagues and collaborators, the study participants for making this study possible. And yeah, and now I'm happy to take questions.
Thanks a lot, Dhriti, for such an insightful talk. So for the question and answer session, you may use the raise the hand button on Zoom or put the question in the chat and I can read it out. Perhaps a question from me, Dhriti, is so for the data that you used, mainly it was generated using the H3 Africa array. So I was wondering whether you noticed any limitations. Maybe was the array sufficient to for the data that you used, or did you notice any limitations that could be improved?
No, like David, I don't think I saw any limitation or anything. So I think that this was good enough for our study.
Okay, awesome. Still waiting for some questions in the chat. Perhaps another one from me as we as we wait for the more sophisticated questions from the experts in the group.
No, I think 3 or 4 of them are authors of this paper.
Okay. Yeah, so you talked about the impact of this work on genomics research and especially on GWAS.
Yes.
So I was wondering whether you could just comment more on that and also on how it would impact design of imputation panels.
So like this work, especially when you are like doing candidate gene-based study, so generally what has happened till now is people have been sampling for anywhere and everywhere in South Africa and generally clubbing as Black South Africans or something, and then just doing a candidate gene-based study. So especially this is more crucial for the candidate gene-based studies, because in case of GWAS, you can still do population structure correction, and that will help you a lot. But in case of candidate gene-based studies, there are so few SNPs. I mean, this can lead to totally false positive signals. So, and there have been so many studies so far. So I think it will be very crucial for us to go back and relook at those studies. Yeah. And about imputation, like this particular study, I mean, it has nothing to do with imputation. So.
Yeah. Okay. Great. Yeah. I'm still waiting for some more questions in the chat. Or is there anyone who would like to unmute to ask their question?
Dhriti, it's Chris here. Could we just revisit the issue of differences between gender? I know that there have been quite a lot of differences between the structure, or at least the ethnolinguistic composition of the males and females, for example, in the Soweto group. Do you want to comment on that?
So, Chris, just to understand your question, so are you talking about the sex bias?
Yeah,
that we
sex
observe?
differences between male and female.
Yeah. So, so what we observed, so this was something very common across all the groups here, all the ACB groups. So invariably there has been like, so the plots that I was showing you, it was very complicated, so I didn't go into the details there. So but what all the plots basically indicated is there has been the more Khoe-San female Khoe-San female gene flow into these Southeastern Bantu speaking groups compared to males. And the only thing that we can comment is it was not uniform throughout the different groups. So there were differences in the extent of female bias for the different groups. I think that is the only thing we can comment from this so far. So I don't know if that answers your question, but these are only like, we can't go beyond a certain point of discussion here. Like, if you have any comment, Chris, please go on.
But one of the issues being that for some of the, for example, cancer phenotypes we study, we're looking at female-only cancers like cervical cancer and breast cancer. So then using male and female controls against female-only cases then potentially becomes a problem.
Yeah, that's true.
Something to be aware of.
Sorry, Scott, sorry to interrupt, but would it cause a problem? Because on if you're looking at the admixture is distant enough in the past that on the autosome and on the X chromosome, you wouldn't expect to see such a significant difference, at least on the autosome.
Are you saying this to me, Scott? Discussing with Chris.
Because isn't the difference mainly on we see the difference in the mitochondrial and the Y chromosome. The Y chromosome, we see this distinct pattern rather than in the autosomal regions.
I thought that there's a different ethnolinguistic composition historically of the male and females broadly in the Soweto group. And therefore there will be many SNPs that differ in allele frequency on the autosomes between male and female breeding.
So, Chris, frankly speaking, you didn't look at it. So probably it's a good point to explore.
But I would think that admixture is distant enough in the past that there's enough mixing that you're not going to see that in the autosomal regions. You're only going to see it in those regions that preserve the founding population.
Okay, we should talk about this offline because we have seen problems in our female GWAS cancer when we're using male and female controls.
Yeah, this is Ananyo, just to add that I mean we observed a very similar thing in another study where we were trying to look for ethnolinguistically concordant participants from Soweto, and somehow it turns out that most of the participants who are concordants happen to be males, and so there are very few females who seems to be concordant. So we are actually looking into that data carefully for that study. But Chris, I think this talks to your point that there might be subtle differences in histories of these populations which might bring in mild biases. So I think really before looking into these data sets and with your experience, we are now gaining insights that there are other layers to it which was not a part of this study but which needs to be looked at very carefully. Like for example, we did a similar study with I think Sean and Dhriti and we co-supervised 2 honors students, and we just tried to look at marriage patterns in participants from Soweto, from Agincourt, and we could kind of also get this, like there are certain hidden biases that are there. So that was also like an honest project, so we kind of worked on it for a month and then almost forgot it. But I think as you are pointing out, there are other layers of history perhaps that can be mined from this data, and we will definitely look forward to getting deeper there.
Yeah, thanks a lot, Chris, for raising such an important issue. Just checking if there are any other questions for Dhriti. It seems not. Yeah, so, oh yes, there is a comment from Aiton. Aiton, would you like to unmute and?
Yeah, sorry, I'm actually just trying to understand, follow what the previous point. So if the number of males and females is the same, surely after the first generation, even the frequencies on the autosomes will start tending towards the middle.
Yeah, I don't know. Scott, would you like to just talk to that, maybe?
So, I mean, I would expect that would be the case, but maybe what we're seeing is that there's something subtle happening in, at least in South Africa, and maybe this is still the product of apartheid and migratory labor system, is that you're seeing different social processes that have brought men and women to the cities, and therefore there's a slightly different selection process, but that's just speculation. I would tend to agree with Aiton that if you're just talking of a general population, then we know that what this work shows is that the founding population is different. There's a difference between the men and women founding populations, but I would agree that after one generation, the autosomal frequencies should be the same among men and women, because there's no, well, I don't understand the biology why the men and women would be different in the autosomal regions.
Unless the number of males is very different from the number of females.
But and also we're not talking of one generation, we're probably talking of 10 generations. So at least.
Yeah.
So I don't. So I think the sort of thing that Ananya is talking about may well be due to the fact that the populations are different because different things bring different types of people to the cities.
Yeah, that's what we were thinking about. And I guess the data is there. So we just need to run some more analyses of that because we actually have the data in hand.
Yeah. Yeah. I'll let Dhriti have the last word on that. It looks like you had a final thoughts, Dhriti?
So I think everyone, like most of the people here have been through this talk at least once. So I think I hope there is no more questions. So maybe.
Okay, cool, awesome. So thanks a lot, Dhriti, for sharing with us this impressive work, and thanks for everyone that has attended the webinar. And Dhriti, we hope that you're keeping safe and well amid all the challenges posed by the pandemic. And I hope everyone that has attended the call is also doing well.
Absolutely.
So,
Definitely.
Awesome.
So I think in India, it's like on the what do I say, like, so the number, if you compare to other countries, it's still like 40,000, but compared to what we had, it's going really, it's doing better. So I think we are now just waiting for the third wave. Yeah.
At least things are good to hear that things are moving in the right direction. So the recording for the webinar will be available on the YouTube channel of the African Society for Bioinformatics and Computational Biology. And thanks again for sparing this time, and hope to see you in the next webinar next month.
And thanks everyone for patiently sitting through the talk and this discussion session.